Today, we introduce FUJIFILM Wako reagents used in pesticide residue analysis. Pesticides are an indispensable product for growing the crops we eat. For example, when growing apples, pesticides are used to mitigate various risk factors. Herbicides can prevent weeds from depriving the soil of nutrients. Insecticides and rodenticides can be used to protect crops from pests and rats. And fungicides are used to prevent rot of fruits by bacteria and viruses. In this way, pesticides are indispensable for growing crops. However, due to their toxicity, if they are absorbed in excess, they can have an adverse effect on the human body. Therefore, governments have set standards outlining the amount of residual pesticide found in food products for human consumption. Japanese pesticide residue regulations are regulated and managed by the Food Sanitation Law's Positive List System and Tap Water Quality Act. The Positive List System prohibits the sale of foods that contain an excess of 0.01 ppm for pesticides, feed additives, and veterinary drugs. With the introduction of this system in 2003, it has become possible to regulate foods that could not be previously regulated, due to using unregistered pesticides which has no residual standard values. In addition, food health impact assessments are still being systematically promoted for pesticides to which uniform standard values have been applied under this system, and the residual standard values are being reviewed in sequence after deliberation by the Pharmaceutical Affairs and Food Sanitation Council. Pesticides are also regulated in tap water. Pesticides in Japanese Water Quality Act are classified as complementary items to be noted in water quality management. Among them, the target pesticides, pesticides requiring examination, other pesticides, and excluded pesticides are classified according to the detection frequency, and these classifications are being revised sequentially based on the test results of Japanese water companies and the latest scientific knowledge. In addition, the Ministry of Health, Labor and Welfare has issued a simultaneous test method for these pesticides by GC or HPLC. From here, we would like to introduce our reagents used in the analysis of residual pesticides. The procedure for pesticide residue analysis can be broadly divided into pretreatment and analysis. The pretreatment process involves solid phase extraction method using a HPLC column, the Quecker's method, and the solvent extraction method. The analysis process primarily includes the instrumental analysis methods using HPLC and GC. Today, we will introduce our products that can be used in these work processes in order. First of all, we would like to introduce FUJIFILM Wako standard products and mixture standard solutions. FUJIFILM Wako carries our trademark TraceSure and Traceable reference material, which are SI traceable reference materials. We also carry analytical standards for pesticide residue analysis that are assured by FUJIFILM Wako strict original specifications. First, we would like to introduce the analytical standard for pesticide residue analysis. FUJIFILM Wako has about 850 analytical standards and mixture standard solutions that can be used for pesticide residue analysis. To manufacture highly reliable analytical standards, we carry out various efforts for quality. We will explain the process flow outlining how FUJIFILM Wako analytical standards are made. After manufacturing raw materials, we first investigate the physical properties of the compound. Physical properties such as volatility, hygroscopicity, and solubility, and these properties have an influence on the test results. It is important to know the physical properties of the compound in order to select the correct test method and obtain reliable and repeatable results. Next, we set an appropriate specification from the information obtained from the physical property survey. FUJIFILM Wako mainly uses GC, HPLC, and quantitative NMR as the test methods for content specification. Here, we would like to introduce the content specification by quantitative NMR method, which is a feature of our analytical standards. Chromatographic methods such as GC and HPLC detect based on the characteristics of the compound, such as absorbance, refractive index, and fluorescence. But the qNMR method detects based on the atomic nuclei that make up the compound. Therefore, in the chromatographic method, one analytical standard can quantify only one analyte having the same characteristics. But in the qNMR method, one analytical standard can quantify all compounds which has a reference nucleus, such as proton and carbon-13. We will explain benefits by qNMR, based on a concrete example. Here is an HPLC chart of an epigallocatechin, which is one of the catechins contained in tea. In the HPLC method, detection is performed based on the absorbance of the compound. Therefore, there were no impurities which has the same absorbance as epigallocatechin. As a result, the content of epigallocatechin was 100%. On the other hand, when the same epigallocatechin was measured by qNMR, the epigallocatechin content was 95.7%. Why are the contents different? From qNMR chart, you can see 0.6% of the signal of ethanol, which is the residual solvent in the epigallocatechin. In the qNMR method, any compounds which have a reference atomic nucleus can be detected. Therefore, residual solvents and the like that cannot be detected by the chromatographic method can be used. As a result, the qNMR method can evaluate the content closer to the true content value, excluding impurities. Due to these characteristics, qNMR method has been posted in various official documents such as the Japanese Pharmacopoeia, US Pharmacopoeia, and ISO at CD 24583. FUJIFILM Wako uses the qNMR method for content specification in about 500 analytical standards. Let's return to the flowchart. After suitable specifications are set through measurements such as qNMR as described in the previous slide, suitable packaging material is selected based on the physical properties of the raw materials. For example, FUJIFILM Wako applies containers that have been confirmed to have low adsorptivity for products that have been confirmed to exhibit adsorptivity in physical property surveys. Finally, we set a suitable expiration date for the product based on physical properties and stability data. In this way, FUJIFILM Wako analytical standards have suitable specifications and expiration dates set according to the compound, so you can use them with confidence. In addition, FUJIFILM Wako has attached a Wako product manual to all analytical standards. We will briefly explain the contents of the Wako product manual. On the left side, compound information such as chemical name, CAS number, structural formula, and content value that can be used as reference information for analysis. In addition, handling instructions developed according to the physical properties of the product are described, so that correct results can be obtained even in your measurement environment. On the right side, you will find our measurement conditions and a chromatographic chart that can be used as a reference for studying analytical conditions. We would like to support your analytical environment not only with the supply of analytical standards but also after purchase through Wako product manuals. Next, we would like to explain the SI Traceable reference materials TraceSure and TRM. Before introducing the standard substances, we would like to explain about SI traceability. SI refers to an internationally determined unit. SI is composed of 7 base units: length meter, mass kilogram, time second, electric current ampere, thermodynamic temperature Kelvin, amount of substance mol, and luminous intensity candela. Before SI was used, various units were used in each country and field, which caused confusion as globalization progressed. Therefore, SI was born to unify the units of the whole world. As a result, these common units have been recognized worldwide, and smooth commercial transactions have become possible. SI traceable in terms of reference standards means that the measured value is connected to SI, and the measured value is reliable and compatible. As an example, the SI traceable system diagram of Traceable is shown. Traceable reagents are produced after the calibration by NMIJ, National Metrology Institute of Japan, or SERI, Chemicals Evaluation Research Institute, which are designated calibration institutions by the Minister of Economy, Trade and Industry Japan. In addition, their calibration methods are SI traceable methods since the calibration values are connected to SI units. From this system diagram, it can be said that the Traceable is an SI traceable reference material. In other words, using SI traceable reference material means that reliable value is assured by connecting a measurement result to an SI unit. By building an SI traceable system, of reference materials are manufactured within the system, we can assure the exact purity of them. We will introduce FUJIFILM Wako SI traceable reference materials. Traceable and TRM are both SI traceable reference materials, except that they have acquired certification. Traceable is manufactured under accreditation by IA Japan, an accreditation institute that is a member of Mutual Recognition Agreement. An IA Japan certificate is attached to the Traceable product and the purity value is internationally accepted. Here is an example of an IA Japan certificate. The certificate clearly states the IA Japan logo, which indicates that the product is manufactured by a company certified by IA Japan, and also states that the certified purity value of the reference material is internationally accepted. TRM products are manufactured to the same high standards as Traceable using SI traceable material calibrated by NMIJ or SERI. However, TRM products have yet to acquire certification from an accreditation body. However, Wako original certificate is attached to the TRM. This certificate states the purity value and uncertainties of the reference material, and states that it was calibrated by an SI traceable method. As a supplement, we will explain the traceability system diagram of Traceable. We mentioned in the previous slide that Traceable is a reference material certified by IA Japan. As shown in this figure, Traceable is connected to SI units through calibration results from NMIJ or SERI. In addition, they employ two SI traceable calibration methods according to the compound. In addition, Traceable are accompanied by an IA Japan certificate bearing the IA Japan logo, which officially proves that the purity value is SI traceable. In addition, since IA Japan has signed a mutual recognition agreement called MRA, which is a mechanism to confirm that the accreditation programs are equivalent among accreditation bodies in each country, the purity values of Traceable are internationally accepted. On the other hand, TRM is an SI traceable reference material that is calibrated by NMIJ or SERI, like the Traceable, but has not been certified by an accreditation body. Therefore, Wako original certificate is attached to the product. Next, we would like to introduce our mixture standard solution. FUJIFILM Wako has about 40 kinds of pesticides, veterinary drugs mixture standard solutions. We would like to explain the benefit of mixture standard solution manufactured by FUJIFILM Wako. When you are trying to prepare a mixture standard solution, one must consider various factors in order to prepare an accurate mixture standard solution. For example, the value of the balance used for liquid preparation may vary due to differences in the measurement environment, such as temperature, humidity, calibration frequency, and grade of the balance. There is an inherent risk of operational error when an inexperienced technician is performing the analyses. Moreover, it takes considerable effort and expense to frequently calibrate the equipment, purchase analytical standards, and prepare the liquid. FUJIFILM Wako has acquired a wealth of knowledge and experience having worked with the aforementioned public institutions to certify our reference materials. We are experienced with manufacturing analytical standards under ISO 17034 and 17025 and performing liquid preparation in the appropriate environment. In addition, FUJIFILM Wako issues specifications and COA that assure the concentration value of the mixture standard solution. By using FUJIFILM Wako mixture standard solutions, one can streamline the liquid preparation process and still perform quick and accurate analyses. FUJIFILM Wako provides about 40 kinds of pesticide and veterinary drug mixture standard solutions. FUJIFILM Wako has launched a search site that allows you to search for these products more efficiently. Before now, when searching for a mixture standard solution, you needed to search from our website or catalog, or consult with our sales team or our distributors. These search methods sometimes took a long time to find the product, and pesticides with multiple aliases were omitted from the search. FUJIFILM Wako search system for mixture standard solutions of pesticides veterinary drugs is a new search engine that solves these problems and allows you to search for mixture standard solutions from the analytes to be inspected. Usage is simple. First, input the analyte name in the left-hand search box. After inputting the partial name of the analyte, you can select keywords from the available keyword list. Some pesticides may have multiple aliases, but this system can search any name. From the search by CAS number on the right side, you can search by the CAS number of the analyte. FUJIFILM Wako also sells analytical standards as salt, but you can search for both CAS number salt and free base. If you cannot find the analyte, you can select the analyte from the list by clicking Search from Analyte List. If you want to delete the registered keywords, click the X mark on the right. If you want to initialize the registered keywords, click Clear All. After registering all the desired keywords, click Search. Search results are displayed in descending order of degree of matching of registered keywords. Each product has a mixture standard solution and single component icon. Click the product name to go to the product page. Circle 4 shows a list of components contained in the mixture standard solution, and registered keywords are highlighted in pink. Clicking each component will take you to the single analytical standard product page. You can switch the analyte, CAS number, and alias display by clicking the tab. As related information, HPLC columns recommended by FUJIFILM Wako and related official test methods are displayed. As an example, we would like to introduce the Pesticides Mixture Standard Solution WQ Series. FUJIFILM Wako has Pesticides Mixture Standard Solutions that applied for simultaneous analysis methods in Japanese Water Quality Act. In addition, our mixture standard solution corresponds to the physical properties of each compound. As an example, iminoctadine, diquat, and paraquat have the properties that they are highly adsorbed on the wall surface of glass appliances, and the recovery rate is significantly reduced. Therefore, polypropylene is recommended for all instruments that contact with the solution of them in the Japanese Water Quality Act. FUJIFILM Wako uses polypropylene containers for the iminoctadine, diquat, and paraquat mixture standard solution WQ7. So, you can use it with confidence. Next, we would like to introduce FUJIFILM Wako HPLC columns and SPE columns. First, we would like to introduce the solid phase extraction SPE column Presep Agri. This column is a SPE column specialized for the pretreatment of samples containing residual pesticides. Presep Agri is available in two types, a cartridge type and a syringe type. This is an example of spike recovery test of 31 pesticides using Presep Agri. As you can see from this table, Presep Agri has both hydrophilic and reverse phase characteristics, so it can be used not only for highly polar components, but also for hydrophobic components such as MPP, teurum, and NAC, and metal coordinating pesticides such as ashram and oxine copper, which are difficult to recover with a reverse phase SPE column. Next, we would like to introduce FUJIFILM Wako HPLC columns. FUJIFILM Wako offers a wide range of HPLC columns for pesticide residue analysis. We recommend these 4 series as the main HPLC columns for pesticide residue analysis. WakoPak WakoSil 2HG has the highest number of theoretical plates among our ODS columns, and is used in quality tests for many of our analytical standards. WakoPak Ultra can be used under highly basic and acidic conditions, making it suitable for the analysis of analytes that are difficult to separate at neutral pH. In addition, since WakoPak Ultra can be used under high-pressure conditions, it can also be used in HPLC, and it is possible to short the analysis time. WakoPak Navi has packing agents with various carbon chain lengths from normal ODS to Triaconta silica gel. WakoPak Navi is effective for congener analysis because it exhibits specific 3-dimensional structure recognition ability by increasing the chain length. WakoPak WakoSil Agri 9 is a column specialized for the pesticide residue analysis. WakoPak WakoSil Agri 9 assures the separation of 9 pesticides simultaneously, including difficult-to-analyze pesticides such as osmium and oxine copper, by combining with a dedicated eluent. Analysis examples with these columns are posted in the Wako product manuals attached to the analytical standards and in the search system for mixture standard solutions of pesticides, veterinary drugs, we mentioned earlier, so please refer to them. FUJIFILM Wako also offers demo columns, so please feel free to contact us. Finally, we would like to introduce solvents for residual pesticide analysis. When selecting an analytical solvent, it is important to use a solvent whose quality is properly assured for the purpose. For example, when analyzing a sample by dissolving it in a solvent, if a solvent whose impurities are not assured is used, even if an impurity is confirmed on the chromatogram, it cannot be determined whether the impurity is derived from the sample or the solvent. As the result, it is difficult to obtain accurate results without high-quality solvents. That's why FUJIFILM Wako has a range of solvents that are assured for different analytical methods. We have summarized FUJIFILM Wako solvents for LC and GC, which are mainly used in pesticide residue analysis. HPLC and LC/MS are methods of detecting the absorbance and fluorescence of the analyte after dissolving the sample in a solution and separating it in a column. Therefore, the solvent used in LC must contain few impurities at the absorbance and fluorescence wavelengths to be measured. FUJIFILM Wako solvents for HPLC and LC/MS assure impurities below standard at specific absorbances, making them suitable for LC analysis. In GC, the sample solution is thermally vaporized, column separated, and then the electrical signal of the analyte is detected. Therefore, the solvent used for GC is required to contain few impurities in the vaporized state. FUJIFILM Wako concentration 300 or 5000 series assures impurities below standard when the solvent is concentrated 300 or 5000 times, making them suitable for GC measurements. We will explain each solvent in detail. HPLC solvents are assured to have impurities below standard at specific absorbance and fluorescence in order to prevent loss of sensitivity due to background noise. In addition, FUJIFILM Wako performs a gradient test to assure low background noise even under gradient conditions, and assures low peroxide amount to prevent sample decomposition. Chromatograms of acetonitrile for HPLC grade and general grade are shown here. Compared to the general grade, the HPLC grade has fewer impurities at specific absorption wavelengths, making it suitable for HPLC analysis. Solvents for LC/MS are assured for mass spectrometry in addition to the assured items for HPLC. Specifically, we guarantee low background noise on mass spectrometers and assure particles to prevent clogging of capillaries at connections. This is the baseline change when using solvents for HPLC and LC/MS. The LC/MS grade assure low background noise on the mass spectrometer and shows less baseline change than the HPLC grade. Assured items for general grade, for HPLC and LC, MS solvents are listed in the table. As you can see in this table, HPLC grade assures peroxides and impurities at specific absorbances are below standard, and performs gradient tests that assure low background noise under gradient conditions, compared to general grade. LC, MS grade assures analytical suitability for mass spectrometers and particles in addition to the assured items for HPLC grade. The concentration 300 or 5000 series is a solvent that is assured to have impurities below a certain level at 300 or 5000 times concentration, making it suitable for pesticide residue PCB analytical tests. Therefore, concentration 300 or 5000 series is suitable for pretreatment of samples that involve concentration and solvent for prepare the GC samples that involve thermal vaporization. We have summarized the assured items for the concentration 300 or 5000 series. In addition to assured items such as water, residue after evaporation, and content, concentration 300 or 5000 series has impurity peaks at 300 times or 5000 times concentration are assured to be half of the pesticide fluorine of BHC 20 nanogram in ECD detection, and to be 1/5 of the pesticide MPP 0.5 nanogram in FPD detection by performing suitability for pesticide residue PCB analysis test. FUJIFILM Wako website provides information that can be used for residual pesticide analysis and the latest information. Please search for Wako pesticide. FUJIFILM, value from innovation.